back after the break. My name is Michelle, and it's my pleasure to introduce Arnaud Gautier, who was so kind to jump in for Catherine Lang and give his talk today instead of Friday. A few words on Arnaud. So he received his PhD in 2005 from the INS in Lyon, and he then moved to the lab of Kai Johnson in Lausanne, which is actually also my lab currently. After his postdoc with the group of Kai Johnson, he went to the UK and did a one-year postdoc with Jason Chin, where he worked on photocontrol of proteins mostly. And after that, he moved to Paris, where since he has been an independent investigator, where he works in the lab of biophysical chemistry and is mostly working on this interface between biology and chemistry, and tries to make use of this semi-synthetic approach that we've already seen a little bit this morning. He's also the recipient of the Médaille Bronze of the CNRS in 2017, if I'm not mistaken. And he's also received an ERC Consolidator grant in 2016. And we're looking forward to your talk.
Okay, so now it's working. So again, thank you very much for the introduction. Thank you very much for the invitation. I'm very glad to be here today. I would have loved to attend such a meeting as a student or postdoc. And so today what I will present you, it's a bit in the continuation of what Kai Johnson introduced you this morning, but how we can combine protein engineering and chemistry to develop new fluorescent probes for biological imaging and try to combine these chemical and genetic approaches. But really, before starting, really to show you what I mean by inducible chemical and genetic fluorescent markers, I just wanted to come back to two categories of fluorescent markers or probes, which are fluorophores and fluorogens. And it's two words that doesn't mean exactly the same thing. And as I will use both during this talk, I just want to remind you slightly just the definition using the etymology of these two words. So fluorophore, just a compound that literally bears fluorescence, and it's always fluorescent. And so you have mainly two big categories of fluorophores, organic fluorophores, as probably you've seen this chromophore yesterday in Lux Levy's talk. And organic fluorophores are really very interesting because they can be super bright, very photostable. And you can use chemistry to change their function. You have a certain versatility. You can really try to just by changing slightly the chemistry, change and tune their function, their either their color, their brightness, their photostability. Of course, one of the issues with the organic fluorophores is that it's quite difficult to really have a selective leveling inside cells. So we saw this morning approaches to try to circumvent this problem. It's too close to my, like this. Like this, maybe it's better. Technology. Yeah, I will try like this. Is that okay? Okay. Yeah. So what I wanted to say, it's yeah. It's a bit louder. It's that yes, with organic fluorophores, you have this problem of selectivity. You need to couple them with substrates to try to have a kind of selectivity and to couple them with tags. So one way to circumvent this selectivity issue is to use genetically encoded fluorophores. So you've heard yesterday as well about fluorescent proteins. So the beauty of these fluorescent proteins is that the chromophore itself, it's fully genetically encoded inside their sequence because of these three residues that can cyclize together to form the chromophore after a dehydration reaction and an oxidation reaction. And this really provides you a way to have an absolute selectivity for tagging, because you can fuse the gene of this protein to the gene coding for your protein of interest, and you have a fusion protein. Of course, even though it's a very great tool, it also has some limitations. Here it's just few limitations of this fluorescent protein. I think it's more annoying for you than for me. It's first, the size of these proteins are rather large. They're around 25 and 30 kilodalton. They also have for some of them, tendency to oligomerize. So it might interfere with the function of this of the proteins you fuse them. And also this very nice maturation process also that takes some time. Thank you. Yeah, maybe it's better like this. So yes, so as I was saying, so you have this very nice multi-step process to have the maturation of the chromophore, but it takes some time. And as you probably have heard yesterday, it takes between a few tens of minutes up to several hours to fully get synthesized. And this, it's a limitation because if you want to study very fast processes where you would like to follow, for example, protein synthesis in real time following the fluorescence of this protein, you have a delay between the time the protein is synthesized and fully folded and the time the protein becomes fluorescent. And the last limitation of GFP is also that it's strongly oxygen dependent. If you are not in presence of oxygen, molecular oxygen, these proteins are not fluorescent because of the last step of the maturation process, which require molecular oxygen. So this can be problematic. Is it good? It sounds better. So hopefully it should work until the end of the talk. Maybe the end of the slide. Yeah. And so yeah, so I was just mentioning these few limitations of these very nice fluorescent proteins otherwise. So this was the two big categories of fluorophores: organic synthetic fluorophores and genetically encoded fluorophores. And so you also have some probes that are not fully qualified as fluorophores, but that are called fluorogen. And etymologically, fluorogens, if you use the suffix gen, it means that it can produce fluorescence, which means that it's not always fluorescent, but only upon either interaction or reaction with something, it becomes fluorescent. So this suffix gen, you find it in many words like hallucinogen, hydrogen. It's the same idea. So this morning we've heard about a very nice class of fluorogen or fluorogenic chromophore, which are the silicon rhodamine dyes, which depending on the polarity of the solution or the environment, can be either fluorescent when they are in the open form or non-fluorescent when they are in the spirolactone forms. But another kind of dyes that you, I think, all know, which are fluorogens, are the DNA stains that you use, like the Hutch dyes. I think all cell biologists in the room probably have used these dyes to stain DNA. What you know is that this chromophore is only fluorescent when it's bind DNA. Otherwise, it's non-fluorescent or weakly fluorescent. And so it's become only fluorescent when it's bound inside the minor groove of DNA. And it's quite interesting, it's now you can have very highly contrasted images like this one without the need for washing the excess of chromophore. You don't need to wash away because the excess of fluorogen is non-fluorescent. Okay. And so how does this work? So we somehow already in the previous talk talk about how things can modify the fluorescence efficiency of chromophore. So to understand how such fluorogenic chromophore works, we need to get back to the different fates of the excited state. So I guess you have seen this diagram several times since yesterday. But so if you look at the excited state, it can de-excite radiatively, so it's fluorescence, and also non-radiatively through internal conversion. And then you can also observe intersystem crossing to go to the triplet state, but all these processes are quite slow processes. So the faster processes are really radiative processes, so fluorescence emission, and non-radiative processes through internal conversion. So most of the chromophore normally just relax light energy through non-radiative processes, and these are in general the fastest process. So this internal conversion, it's for most chromophore the fastest process for the excitation. And this is due to, for fluorogens, it's due to the fact that you have internal motion that can occur inside the protein. You have vibrational de-excitation and so on. And because of this internal rotation and motion, the molecule can exchange the excess of energy with a solvent through kinetic energy. And you just heat the solvent and you exchange the energy through it. So this process is very fast. So if you look at the fluorescence quantum yield formula, which is given here, you understand directly that indeed if this kinetic constant is very high, much larger than the kinetic constant for the de-excitation, the radiative process, the fluorescence quantum yield is low, okay, because here this constant is much higher than all the rest. But now if you take this very same chromophore, but you lock its conformation and you prevent internal rotation, then now the non-radiative process becomes much slower. It becomes much slower, and the kinetics now of the internal conversion and the radiative de-excitation start to be comparable. And if you are in this situation where these two kinetic constants are comparable, now the fluorescence quantum yield can be higher. So that's why a same molecule can be in two different fluorescence states, a dark state and a bright state, depending on the environment it is. And so we decided to try to use this concept of fluorogens and fluorogenic chromophore that can have two different brightness state, a dark state and a bright state, depending if they are free or bound to something, to try to develop inducible chemical genetic fluorescent markers. So the idea is to take a fluorogen which is non-fluorescent in solution, but that could become strongly fluorescent if it's bound specifically to a protein tag. And the idea is really to combine so the tunability and versatility of synthetic molecules together with the ability to target specific proteins inside cell using a genetic tag. So the advantage of using a genetically encoded protein tag is to be able to target the protein where you want, so to fuse it to a protein of interest, to tag a cell or whatever, and also to benefit from their evolvability, eventually to modify their function. So the fluorogen we started to work with is a very small chromophore which has this chemical structure. So it's called hydroxymethylbenzylidene-rhodanine, so HMBR, the shorter name, and it really has the ability to be weakly fluorescent or almost non-fluorescent in solution because of internal rotation around this bond here. So here you can see its fluorescence spectrum when it's free in solution in white, so it's almost flat in this situation. And we could engineer a small protein tag that can bind specifically these fluorogens and activate its fluorescence. And so here you can see the fluorescence of the exact same chromophore when it's bound to the protein. And you can see that you have a large increase of the fluorescence efficiency just upon binding this protein tag. So here there is almost a 3 order of magnitude increase of the fluorescence efficiency between the unbound situation and the bound situation. So in that case, this increase of fluorescence, it's mainly due to an increase of the fluorescence quantum yield upon binding. But it's not only due to this. It's also due to the fact that upon binding, this chromophore undergoes a second spectroscopic change. Its absorption properties and therefore its excitation properties are changed. This chromophore, when it's free in solution, it's mainly protonated and absorbed around 400 nanometer, while when it's bound to the protein fast, it absorbs around 480 nanometer. There is 80 nanometer redshift, which is due to the deprotonation of this phenyl group into a phenolate. And this now allows really to have a system where when you choose your excitation wavelengths, you just excite the bound fluorogen and very little the unbound fluorogen. So this furthermore increases the fluorogenic response of the system. So because of these two spectroscopic changes, we called fast fluorescence activating and absorption shifting tag, just to highlight what this small protein tag does on this fluorogenic chromophore. So this chromophore is very small, it's very simple to synthesize. It's a one-step synthesis from two commercially available compounds. The synthesis can be done by anyone. It's just you mix the two compounds together in water, you heat up, the compound precipitates, and you just have to filtrate somehow. So the synthesis part was the easy part in the development of this system. The difficult part was the design of this protein that binds selectively this chromophore and activate the fluorescence. So why it was difficult initially? First, because this chromophore, it's a non-natural chromophore, so there was no obvious proteins that could bind selectively this guy. So we had to look for proteins that could, upon engineering, bind selectively this chromophore. So we needed a protein with already a cavity that could accept somehow similar chromophore. And so we ended up by discovering this small protein called PYP, which stands for photoactive yellow protein, which is very interesting for developing a tag for several reasons. The first one is that it's a very small protein. It's 14 kilodalton. So if you compare it to GFP, which is 26 or 27 kilodalton, it's almost twice smaller. It's smaller than SnapTag or AlloTag. And what's interesting in our case is that this photoactive yellow protein, so it's not a fluorescent protein, just to be clear. Even though the name could let you think that it's a fluorescent protein, it's not a fluorescent protein. The yellow here, it's about its color, which comes from this chromophore, which is covalently attached to a cysteine within the protein. And this chromophore, it's a para-hydroxycinnamoyl chromophore, which upon light illumination can photoisomerize. And so this photoactive yellow protein, it's actually a photoreceptor, a blue light photoreceptor that you can find in some bacteria. But so what was interesting for us was not really the biological function of this protein, it was really the structure of its natural chromophore. Because when you look at the structure of this chromophore and you look at the structure of our fluorogenic chromophore, they are not very far away. You have a phenyl ring, a double bond, a carbonyl group on both compounds. In terms of size, it's rather the same size. So we thought that the cavity that accepts this chromophore inside the natural PYP, upon some engineering, could probably bind selectively and non-covalently this fluorogenic chromophore. So with a bit of protein engineering, we could indeed develop fast from this PYP sequence. So basically the difference between PYP and FAS is just 9 amino acids. We mutated the cysteine 69, which is normally it's used to covalently attach the natural chromophore of PYP to a glycine, just to make sure that this cysteine wouldn't interfere in any way with our interaction with our fluorogen. And we replaced the loop that normally gate the protein binding cavity, which normally has this sequence, by this new sequence, which is here. And this basically allows to flip over the ring and open the binding cavity, and also probably to make contact with the chromophore with the fluorogenic chromophore. So the question you might ask is, how did we decide to choose this particular sequence? So we couldn't predict it by any means. So we used directed evolution to do this. So the idea here is really to explore a very large sequence space to try to identify mutants that could have the function we are looking for. So we use saturation mutagenesis to basically randomize the sequence of the loop. And we generated almost all the possible sequences. And then we inserted these mutants inside yeast cells, and we expressed the mutant at the surface of the yeast cell. And so now we have a very large yeast cell population where each cell expresses a different variant of PYP. Then you can use this large population of yeast cells at the fluorogen if they express a variant that does interact with the fluorogen and activate its fluorescence, the cell will become fluorescent. Okay. And if it's not the case, the cell will be non-fluorescent. And now then you just need something to keep the fluorescent cells and discard the non-fluorescent cells. So luckily for us, there is a fantastic tool which is a fluorescence activated cell sorting, which is a device that allows to analyze the fluorescence of a single cell at very, very high throughput. So when I say high throughput, it's 50,000 cells per second. And so typically in one day, we can analyze something like almost 1 billion cells. And this allows us to cover the diversity of our yeast cell library, which is typically above 10 or 100 million cells. 10 or 100 million variants, sorry. And so by using these facts, we can basically just analyzing the fluorescence of the cells, just then collect specifically the fluorescent cells. Of course, what we are looking is at very rare events. So we cannot really extract in one single cycle the cell of interest. We need to do iterative round of sorting and amplification to really enrich the population in these fluorescent cells. But after several cycles, we can basically enrich the population in cells that express these fluorescent variants in presence of the fluorogen. We can isolate these cells, extract that DNA, and then obtain their sequences. So just to give you an idea of the difference between PYP and FAST for the binding and activation of HMBR, the fluorogen. Here it's the fluorescence of a monoclonal cell population expressing either PYP on the left or FAST on the right. In absence of HMBR in red and in presence of HMBR in blue. So if you look at cells expressing PYP, basically there is almost no difference in terms of fluorescence. If you add HMBR, there is really just a slight shift, which is almost undetectable. But now if you take cells expressing FAST in presence of this fluorogenic chromophore HMBR, you have a 3 order of magnitude increase of the fluorescence of the cell population. And this shows you the activation of the fluorescence of this chromophore. So of course you can analyze by FACS. I think you are mostly interested by imaging, so you can also analyze it by fluorescence microscopy. And so here it's exactly the same yeast cells expressing either FAST or PYP, both in presence of HMBR. And you can see that in the case of FAST, you have a nice cell surface staining, as you would expect, while in the case of PYP, you do not see any fluorescence in the corresponding channel. So now we have a system really that is very comparable to what Kai Johnson introduced you this morning. We have a protein tag that we can fuse to a protein of interest just by using DNA technology. We can express this fusion gene, have a fast tag protein, add a fluorogen, and instantaneously image without any need for washing or anything. So you can reduce some kind of labeling on demand really rapidly. So here it's mammalian cells. We show that we could really use FAST to label proteins in various systems. So here are just a couple of examples. So here it's the yeast cell I showed you before. We showed that we could label fast tag proteins inside bacteria as well. It works also, of course, in mammalian cells in different cellular localizations. So you can fuse FAST to various proteins, and just by addition of the fluorogen without any washing, image with very high contrast. So here just a couple of examples where you have H2B fused to FAST, so it's in the nucleus. FAST fused to a membrane targeting sequence, lin 11. So you see the inner membrane here. You can target FAST to the mitochondria, also to proteins that can bind, for example, the cytoskeleton, like this is ensconcing proteins that bind microtubules. So here it's, when we tested the mammalian cells, was mainly in quite robust cells, HeLa cells or egg cells. We also showed that we could use FAST to label proteins in maybe more delicate cellular systems, like live neurons. So here what you see, it's the dendrite of a neuron, and the bright spots are inhibitory synapses that accumulate a protein called gefferin, which is a scaffolding protein for neurotransmitter receptors. And here it's just two experiments where we co-transfected either gefferin fused to FAST or gefferin fused to M-cerulean, which is a blue fluorescent proteins. I think you can see that we have a perfect co-localization of the two proteins. So here what I show you so far, it's mainly it was only one photon microscopy. We also recently showed that we could use FAST to do two photon microscopy. It seems that the the probe has a efficient 2-photon cross-section. So here it's just an experiment where we, with some collaborators, we tagged measles viruses with FAST, and we infected virus cells with this tagged virus, and we could image them by 2-photon microscopy. So the idea is, by using 2-photon microscopy, it's then maybe to be able to do similar experiments in multicellular organisms like mice or whatever. So the question, of course, if you start to think about using such system in more complicated system like only cells, like multicellular organisms, is it really doable? As Kai mentioned this morning, it's sometimes quite difficult to make the transition from cells to multicellular organisms, especially when you use small molecules that need to diffuse across a multicellular system. One interesting thing when you use fluorogen, it's however that the fluorogenic behavior normally helps you a lot to do labeling inside a complex multicellular organism, because as you don't need to wash to have a high contrast, the fluorogen can be present all the time, and you can really have a good contrast even in a multicellular system. So to show this, we just collaborated with people working in the zebrafish biology field, and we just co-transfected the red fluorescent protein mCherry and FAST inside a zebrafish embryo. So here what you see, it's a zebrafish embryo of 1 day. And I think you can see that the expression pattern of the two proteins fully overlap. So we could really show that we can efficiently label proteins with this system in zebrafish embryo, and that the small molecule really can diffuse across the entire embryo very rapidly. So 1 day it's still a stage of the embryogenesis where the fish is quite permanent. We haven't tried at later stage, so maybe when the skin of the fish start to really grow and become very thick, it might be more problematic, but at least at this stage, it's very quick. And when I say very quick, here it's written 30 minutes, so it's more or less the time you need to have complete labeling inside the zebrafish. So here just the movie showing in real time, almost real time, accelerated. The diffusion of this, the labeling of FAST inside this zebrafish. So within almost 30 minutes, it's almost finished. And so if you compare it, I don't know if some people in the audience do staining inside zebrafish with antibody or even with SnapTag or AlloTag. With antibody, we need several days to have a good staining inside an embryo. And I guess with SnapTag or AlloTag, you need probably several hours to really have an efficient labeling. So here, with only a few tenths of minutes, we can really have something which is complete. And this is due only to the fact that the chromophore we use is very cell permeant, and it's very small. And size in this case is really important, and it's the reason why it's so efficient to cross membrane. So a very interesting feature of FAST, it's that the binding of the fluorogen, or the association with the fluorogen, is fully reversible. And this reversibility so no, it's not that I want to say, yeah. So it's fully reversible. I will come back to this afterwards. But at least, so you have this dynamic exchange. You can characterize the on-rate association constant and the off-rate dissociation constant. And from these two parameters, you can basically calculate the half-time for the labeling process, which is given by this equation. And Of course, it depends on the concentration of the fluorogen, but if you choose a concentration which allow to have full labeling of the protein, from this kinetic constant, basically what you can calculate is a half-time of binding of 10 milliseconds. Okay. So as soon as you mix the two components within 10 milliseconds, if you if you have no diffusion, if you don't need diffusion for the association, it's finished. So again, if you compare it to covalent labeling, this is several order of magnitude faster than SnapTag or AlloTag. And so just to give you an idea of how quick it is, you can just mix a solution of fast and a solution of the fluorogen under blue light illumination, and what you see, it's instantaneously it's fluorescent. Okay, there is no I mean, by eye you cannot see. It's almost instantaneously fluorescent. So now, if you compare such system with autofluorescent protein, where I told you that you have this maturation process, and the maturation of the chromophore takes some time after the synthesis and folding of the protein to synthesize the chromophore here. Provided that the fluorogen is present, as soon as the protein is synthesized and folded, it is fluorescent. Okay. So to show this, what you can do is just to fuse fast, for example, to a red fluorescent protein like M-cherry, and express this fusion in an in vitro expression system, for example, and follow the fluorescence of fast and M-cherry. And what you see, it's even though you synthesize a single protein, you do not have the same fluorescence profile. What you observe is that the fluorescence of M-cherry appears almost 1 hour after the fluorescence of fast. And here the fluorescence of fast is very close to the actual synthesis rate of the protein. So here you can use such system really to monitor in real time fast processes like protein synthesis. So another interesting feature you can extract from the kinetics parameter of the binding, it's the residence time of the chromophore inside the protein. So the residence time of the chromophore inside the protein is reciprocal of the off-rate kinetic constant. And it gives us something around 160 milliseconds. So it's a quite short residence time. So what does it mean, so short residence time? It means that if you're at steady state, you have very highly dynamic binding. It's the system is continuously binding and unbinding. Okay. So this, if you look at single molecule, in theoretically it's a blinking system. Okay, it's either it's on or either it's off. So we think that maybe this kind of system could be used as a blinking probe for super resolution microscopy. We've heard this morning DNA paint. Somehow this system could probably be used for doing protein paint and do super resolution microscopy. So we haven't any data on this, though I cannot really fully demonstrate that it would work, but at least such system with fast binding dynamics probably a good way to be able to do protein paint in live systems. A more interesting also feature of this very short residence time is that basically if you are at steady state and you wash away with the fluorogen, normally you should dissociate very rapidly the complex. Okay, it should take less than 1 second to dissociate. And this could be used really to switch off easily fluorescence when you need to switch it off. So to demonstrate this, we just took cells expressing fast in the cytoplasm. We put them in a microfluidic device that allow us basically to inject a washing solution or a staining solution containing the fluorogen. And what you see here on this movie is that within few seconds, as soon as you add HMBR, you have almost full labeling. And as soon as you remove it, it dissociates also very fast. So here it's the time is in seconds. So it takes roughly 20 seconds to get fully labeled inside cells, and roughly 40 seconds to get unlabeled inside cell. And here, what limits the rate of the labeling process is just the diffusion of the molecule across the cell membrane and within the cell. Okay. But it's still already very quick. So what's really interesting with a system that is composed of a protein part and a small synthetic molecule is that you can start to play with both parts to eventually change their function or their properties. So an easy thing to do is just to change the structure of the chromophore. So what can happen if we change the structure of the fluorogen? So what we did is just to modify the substituent on the aromatic ring just by introducing two methyl group or methoxy group or two methoxy group on this aromatic ring. And what we could observe is that we could still form very fluorescent complex with fast, and especially the two yellow ones here could increase the fluorescence performance of the fluorescence complex. So with HMBR fast, it's around 25% fluorescence quantum yield, but with this HBr35DM with two methyl group now, it's raised up to 50% fluorescence quantum yield. And this you can easily understand it because, as I told you, the fluorescence increases due to immobilization of the fluorogen inside the protein cavity. So if you increase if you put two handles here on the aromatic ring, basically you prevent motion. More interestingly, to my opinion, in this study was the discovery that by introducing two methoxy group, we could basically change the spectroscopic properties of the complex. Normally fast with HMBR absorb around 480 nanometer and emit around 540 nanometer. But now with this orange dye HBr35DOM, it absorbs around 520 nanometer and emits around 600 nanometer. So it's almost red, it's orange red, and so you can start to play with colors now. So this different fluorogen really works as efficiently as HMBR to stain fast tag proteins. So here just a couple of examples where we just stain fast in the cytoplasm, in the nucleus, or in mitochondria with these these three different fluorogens, and you see really identical behaviors. So I told you that we can change the dye, but we can also, of course, engineer the protein part. And recently we could isolate, so it's the work of Alison within the audience, together with Federico Pimenta, isolate some improved fast variants that now allow us to reach almost 60% fluorescence quantum yield for most of the chromophore, and that start really to have systems which are comparable with the best fluorescent proteins in terms of fluorescence efficiency. Of course, we are still far away from the Janelia chromophore from Luke Levis, but we are still already quite bright for such system. So here it's basically the spectroscopic properties we have right now in hand, or what we published. The question is whether with similar strategy we could increase the number of colors we can really obtain. So currently in the lab, so Ella within the audience, starting her PhD, and Chen as a postdoc, they are trying to really increase the palette of color we are doing. We can obtain with analogous systems. So I will not show you any structure because of intellectual property issue. It's not yet patented. But what I can tell you is that we should soon be able to have all these different spectroscopic properties. So we should be able to go up to 700 nanometers in terms of wavelengths in the near infrared, and go down to 460 nanometers on the blue side. So with this family of chromophore and this family of protein tag, we should be able to engineer a new palette of tag with different colors. So, as I told you, so we have a single tag, and now we can change its color. It can be green. If we use HMBR, it can be red or orange-red. If we use HBR35DOM, so we have one tag, but we can do two colors. And this provides you really with versatility that you do not encounter with fluorescent proteins. You can now really, if you want to use fast in cell lines that already express red fluorescent protein, you can use HMBR to make it green. But now if you want to use fast in a cell line that already express, for example, a green fluorescent protein, you can use this reddish fluorogen to make fast orange-red. And so it's the kind of versatility that you can have with SnapTag and AlloTag, and you also can have it with such a non-covalent fluorogenic tag. So this is very nice. You can choose the color you want. But you can even do in the same experiments the switch of these two colors. So you can, for example, stain, so it's H2B fused to fast, so in the nucleus, with the red fluorogen. And just by adding an excess of the green fluorogen, you can exchange the color within a few seconds. So you just switch the color. So it makes a nice movie. I really like this movie. I always compare it to a kind of cellular traffic light. Is it useful? It was the question, whether it's really useful. So when Chen, who did these experiments, came to me, I was not sure if it was of any use for imaging. But then I came to see Ludovic Julien, who's in my department, and was very fond of using kinetic properties to do selective imaging. And he told me directly, of course, with such movie, you can selectively extract the signal of fast, even if you have red fluorescent proteins or green fluorescent proteins together present. Okay, because of course, if you look at the green and red fluorescent signals of fast during the exchange, they are fully anti-correlated. It's either the protein is green or it's red. Okay, it cannot be both because the fluorogen compete for the same binding site. Okay, so it's really a unique kinetic signature. And so if you are in presence of a green and a red fluorescent protein, where normally even if you have two fluorogens, you cannot discriminate fast from a green and a red fluorescent proteins because of spectral overlaps. Using such kinetic information, in theory, you should be able to extract the signal of fast, because the green fluorescent protein will not have this kind of behavior, and the red fluorescent protein will not have either this kind of behavior. Okay. So now if you have a system where you have three proteins, let's say EGFP at the membrane, cherry in the mitochondria, and fast in the nucleus, what you can do is just take an image before adding the fluorogen to know where are the green and red fluorescent protein. And then just stain fast with one of the fluorogen and exchange it to the other one, record a movie, and just do use a simple trick to measure the degree of anti-correlation of the green and red fluorescent signal using, for example, two-color cross-correlation. And in theory, you should extract without any difficulty the signal of fast, and you should kill the signal of the green and the red fluorescent protein. So does it work? Of course, if I present it, it does work. So it's the actual experiments that I just described. I read GFP at the membrane, cherry in the mitochondria, and fast in the nucleus. So here it's a three-color image using only two detection channels. Okay, just using this trick. So you can imagine to use such system to really increase the number of objects you can image with a limited number of detection channel. So of course, if you can change the structure of the chromophore to change the color, you can also change the structure of the chromophore to eventually change the physicochemical properties of this chromophore. So what I told you is that the fluorogens we use are very cell permeant. They cross membrane like the blink of an eye. But what if you would like to selectively label a membrane protein and cell surface proteins, and not the proteins which is inside cells? So we decided to also try to modify the structure of this fluorogen to render the fluorogen membrane impermeant. And as you may know, the permeancy properties of small molecules are really dependent on their charge. Neutral molecules cross membrane easily, charged molecules already more difficultly. So what we just decided to do is to try to introduce a charge modifying the chromophore as little as possible, adding this acetate group here on the rhodanine head of our fluorogen. And just by doing this, we could still have a fluorogen that binds fast correctly and gives a good fluorescence, but now which is unable to cross the cell membrane. So we're just an experiment showing this, where it's fast fused to M cherry, which is expressed in HeLa cells. And so on the right hand side, it's you see the fluorescence of the cell in the fast channel after incubation with this membrane impermeant fluorogen for 2 hours, and you do not see much of the fluorescence. And by comparison, on the left hand side, it's the fluorescence you get after incubation with the same concentration of the permeant HMBR for only 15 seconds. And you see that in 2 hours you didn't get any obvious signals. So now you can really use this membrane impermeant HBA2A3E to selectively label membrane proteins at the cell surface. And here it's to show you why is it important to eventually to be able to selectively label proteins at the cell surface, because if you take a membrane protein and you label it with HMBR, what you also see, so you see nicely the proteins at the cell surface, but you also see the protein which is in the secretory pathway. And depending on what you want to study, this can pollute your fluorescent signal, and can be even very strong in when you do imaging. So I don't know if some people in the audience do membrane protein imaging, but sometimes you can have very strong signal coming from the secretory pathway. So here with this new fluorogen, you can selectively label membrane proteins. So you can do it very quickly. So here it's even faster than if you need to label proteins inside cells. Of course, you do not have to cross a cell membrane, so you just have are limited by diffusion. And so within less than 10 seconds, you have full labeling of your system. So now you have a system where really you can really develop a very efficient add and read protocols. You just add the fluorogens and you can directly read the fluorescence. And what's interesting now with such technology is that if you want to have an information on the localization of your membrane protein, you don't need any more microscope. You just need to take fluorescence measurements. Okay. And so you can use device that allow you to do analysis with a much higher throughput than microscopy. So for example, flow cytometry. So by just taking by measuring the fluorescence in presence of the membrane impermeant fluorogen, you can have access to the level of cell surface protein, and you can normalize this level by the total amount of protein by labeling with the membrane permeant dye. So here it's just a very simple experiment showing this, where we just took cells expressing a model membrane proteins and treated cells with two drugs, one which is brefeldin A, which inhibits protein secretion, and another one which is cyclohexamide, which inhibits protein synthesis. So what you do observe, if you look only at the cell surface level of the proteins, is that indeed the two drugs lower the amount of proteins at the cell surface. The question is whether this lower cell surface expression is due to inhibition of the protein secretion or inhibition of the protein synthesis, because you cannot really distinguish just by looking at the expression level in that case. But if you look at the total amount of protein, what you see is that brefeldin A do not affect much the total expression level of protein, while cyclohexamide indeed decrease the total amount of protein because you have inhibition of the protein synthesis. And then you can do the ratio of the two, and you get indeed that only brefeldin A can really specifically inhibit protein secretion. And so now you can really imagine to use such simple fluorescent measurements to screen for more complicated systems. Like, for example, you want to look at protein trafficking, and you want to know whether you have a normal traffic or eventually a defective trafficking. And by using a combination of these two fluorogens, you can really determine the cell surface level of the protein and the total level of the protein. And here what we did, it's just we took a dopamine D2 receptor, which can be seen as a GPCR model, and we use one of its mutants, which is known to have a defective trafficking, because of mutation that basically render unstable the protein and misfold the protein inside the secretory pathway. And by using this double staining strategy, we were indeed able to show that these mutants have really a defective traffic. And now we can imagine to use such approach to develop assays to screen for drugs that could restore trafficking. And you could really do high throughput screening with such approach. So we didn't do any high throughput screening so far, but what we at least showed is that we can indeed, using a piperon, which is a pharmacological chaperone, show that we can restore the correct expression level of this mutant just by a simple measurement of the fluorescence. Okay. So to finish my talk, I would like to share now a final story, which it is how we can use such chemical genetic inducible fluorescent marker to design biosensor. So we've heard this morning a lot about biosensor, yesterday evening as well with Robert Campbell talk about biosensor and how fluorescence can be very useful to analyze the change in the concentration of an analyte. So when you think about a fluorogenic system like FAST, where you have a protein interacting with a fluorogen, you are very much interested in trying to say, okay, could I change the affinity of the protein for its fluorogen by the presence of an analyte? Okay, could I increase, for example, the affinity of the protein for its fluorogen by the presence of the analyte, and try to basically condition the binding of the fluorogen to the recognition of a given analyte. And if you are able to do this, really to shift the affinity of the tag for its fluorogen by the presence of the analyte, normally you can choose a concentration of fluorogen where in absence of the analyte you are mainly unbound, so weakly fluorescent, and in presence of the analyte you are mainly mainly bound and highly fluorescent. Okay. And what is quite interesting is that you can somehow evaluate what is the fluorescence increase you can observe upon a change in affinity. So here it's just simulation where we have binding curve theoretical binding curves for the fluorogen if we have two different KDs, that let's say due to the binding of the analyte. And if you take the ratio of these two curves, you get this blue curve, which basically gives you the fluorescence increase somehow that you can expect upon analyte binding. And what you see is somehow that the maximum fluorescence increase you could expect is directly related to the ratio of the two KDs in absence and in presence of the analyte. Of course, here at this maximal value, it's at concentration of fluorogen where you have barely no complex form. But there is here a situation which is these dark windows where you still form a large amount of the complex with the sensor, and you have a large increase of the fluorescence. So together with Alison, we decided that we should look at this idea of using FAST to generate biosensors. And so of course to make FAST sensitive to a given analyte, you cannot use FAST directly. You need to modify it slightly, the structure of FAST, and add and couple it with analyte binding domains. And in that case, you should be able to promote a change in conformation in FAST upon binding a given analyte and increase its affinity for its fluorogen. So we've heard a lot yesterday about calcium sensor design, and so we decided to try to see whether we could use FAST as a fluorescent module inside a calcium sensor. So we stole the design of GCamp and used M13 and calmodulin as the calcium sensing unit, and fused them to not to FAST but to a circular permutation version of FAST, which allow us to have the N and C termini close together for a more efficient coupling upon interaction between M13 and calmodulin. So it really very much looks like the different design Robert Campbell showed you yesterday evening. Luckily for us, we didn't have to really go through the maze yet to go through just by using this simple design without doing any further engineering. We succeeded in designing a system where indeed we had a system where we could increase the affinity for the fluorogen in presence of calcium. So here what you see is the binding curve for the fluorogen in absence of calcium with a square, and in presence of calcium with a circle. And so you see that you can increase the affinity in presence of calcium. What's very nice also with using a system like FAST, it's that we can change its color very easily just by changing the small molecule. So we don't have to re-engineer everything when we want to change color. We just have to change the chromophore, and we observe exactly the same behavior. So here it's the same sensor, this time using this red fluorogen HBr35DOM. And we could also observe basically this increase in affinity for the fluorogen in presence of calcium. So this change in affinity allows us to have a rather correct fluorescence increase upon calcium binding in our system. So we can have, if we choose rightly, the concentration condition for the fluorogen up to 4 or 5-fold increase in fluorescence. And we could also characterize the affinity of this sensor for calcium. And so we are in the 100 nanomolar range, a little bit below. So which is a sensor which have a slightly higher affinity compared to the sensor Robert Campbell talked about yesterday. So it kind of complements the current state of the art in terms of affinity for calcium. And we could of course show that we could use such system to monitor change in calcium concentration in living cells. So on model systems, so it's just HeLa cells expressing our sensor stained either with the green or the red fluorogen, and here it's the picture at low or high calcium concentration, and so it's cells that are stimulated by addition of histamine that trigger the release of calcium from the ear to the cytoplasm, and we can monitor calcium concentration changes over time. So this simple experiment just show that we can use FAST to design biosensors. So we see another movie showing in real time the change of calcium concentration inside cell using this calcium sensor. So here we did it with calcium using well-known calcium binding domains. But as I told you, we didn't have to do any engineering to do it. We just used FAST to these two domains, and it worked rapidly. And the reason why it worked rapidly is because we don't rely exactly on the same mechanism than the GCamp story or the GCO story. We don't have to have any interaction between calmodulin and the fluorescent reporter part, which is the case for the GCamp family. And so we really think that we can really expand this kind of concept to more easily to other kind of sensing domains to design new biosensors. So I will conclude with this. Just one take-home message. It's that I hope I showed you that to design fluorescent markers that take the advantage of both chemistry and genetics to design this inducible system really open a new prospect for both imaging but also for biosensing, and that we can really expect more from this kind of strategy. So I want just to finish with some acknowledgements. So I have the chance to work with very talented students and postdocs. So I want to thank first Arnaud Plamont, who has been essential for the development of FAST. I want to thank Chen, who was a PhD student when she started in the lab, and we developed the new colors and the cell impermeable fluorogens, and which is now taking the development of the far-red and near-infrared fluorescent markers. I want to thank Alison and Federico, who developed the calcium sensors and also new fluorogens, the new improved variant of FAST, and also Ella, who is in the audience and who's just started her PhD and tried to develop this new blue fluorogens. So if you have any question about the calcium sensor, I think Alison has a poster tonight, so you can go and ask her. And I want to thank some of my collaborators, and I will finish here, and thank you very much for your attention, again for the invitation, and I'm ready to take any questions.
Thank you for that talk. I have a question concerning the fast M-cherry maturation experiments that you did. You showed nicely that M-cherry matures slower and you get the increase later. But I noticed in your graph that there was a decrease in the HMBR fluorescence over time, even though M-cherry plateaued, HMBR decreased by quite a lot. Why?
So you have a fusion between the two proteins. So part of this decrease is that you start to have energy transfer between fast and cherry. So you have a decrease of the fluorescence of fast when M-cherry gets matured. So it's one of the reasons. So you have just thread between, at some point, a bit of thread between fast and M-cherry. Okay.
Since it takes tens of seconds to get for the HMBR to get into the cells
Yeah.
through the cell membrane. Do you think it's a way to study the rigidity or something about the cell membrane, or do you think it's too fast?
No, probably. I mean, I already had this question whether we can use somehow fast to monitor indeed change in the membrane rigidity, or as a way at least to monitor transports. In theory, it should be possible, and I don't know whether it's too fast. I don't know if you work on such question and what would be the perfect time domain for you, but I guess it's possible to do it. Especially you can play with. I mean, you can play with diffusion somehow if you want to make it slower. If you would need to make the source of fluorogen far away to have a slower diffusion, that could be something possible as well. So I think we can probably tune a little bit the kinetics of entry if it would be too fast.
Thanks for the talk. I have maybe a naive question. I hope I got it right, but in your zebrafish model, you basically inject your fluorogen.
So we do not inject it, we just put the fluorogen inside the water of the fish.
Okay, and
So it's not injection, it's just in the water.
so do
So
you
for as long as you can put your embryo in solution, it will work normally. So for Drosophila, I think it would be okay. For mice, start to be more complicated. Probably for mice, you will need to micro-inject the dye, either in the blood or directly locally, where you want to inject to image. So we have preliminary results showing that basically local microinjection works. It seems to work, but currently it's a bit too early, maybe to really answer fully your question. But indeed, it's one of the limitations of the system. It's you need to be able to get the dye inside multicellular organisms. But again, what I would say is that it's very cell permeant, and it seems that even micro-injecting in mice, it seems to get through the tissue quite easily. If you inject it in the blood, it seems that it doesn't stay very long. It's 1 or 2 hours, then it gets degraded.
So you showed us this amazing rainbow of colors. So do you have hopes to modify fast to give specificities of different fast by different colors?
So of course we will not have a single tag that binds everything. So what I showed you for the spectra, it's mainly with only fast, but with some new fluorogens, the performance of fluorescence are not good enough for you to do decent imaging. So we need to evolve fast to make them better, but we can still have the fluorescence spectra. But so what we already started to do in part with Alison as well, is to try it for the red and the green fluorogens to have orthogonal systems to do two-color imaging. So this work. But of course, if we have 5 or 10 fluorogens, we will not be able to make them all fully orthogonal. And the same probably would be difficult to have one tag that binds everything very efficiently. That could be very cool to have one tag that can be really from blue to far red. But I think it's a bit optimistic to think that we can do this. And it's the same for the full orthogonality between all the systems. So we will have a system that could be orthogonal. And with the far red, the structure of the fluorogen is quite different from HMBR, so probably they will be intrinsically orthogonal to the original fast. But yeah, it's difficult to to tell you anything about this. What I can tell you is that we can do orthogonal green and red, right? Two colors for the moment. So we don't have the exact crystal structure of the overall complex. We have the crystal structure of the protein itself, but not with the chromophore right now. So when I show you image, it's more cartoon. That we think it's what happened in the binding of the fluorogen. Yeah, so far we have kind of difficulties to do to crystallize the complex because probably of the binding affinity, which is not high enough to really get the crystal of the complex. Because we have this dynamic binding, so but we want this dynamic binding, but we cannot really crystallize the complex. So the signal to background ratio it's very good. The reason is because the chromophore is fluorogenic. But you could indeed imagine that as the fluorescence activation is due to immobilization of the fluorogen, if the fluorogen binds to something inside cell, you could have unspecific fluorescence. One of the reasons why it's the signal to background ratio is so good in our case, it's because we also have this shift in absorption upon binding, which is very specific to FAST, and which probably it's more unlikely to occur with natural biomolecules inside cells. And this makes a difference, I think, compared to other kind of fluorogenic dyes. I mean, at some point you will have some background, and you will anyway, even though the fluorogen is weakly fluorescent, it's still a little bit fluorescent. So depending on the concentration you use and the concentration of the protein you are looking at, of course, at some point the background could be detectable. But it will be more the background of the fluorogen itself, free in solution, than really any kind of interaction with intracellular components. But you have to test and to check in your condition whether it's a problem. Yeah. You're probably right. I mean, we don't do any calculation, and it's maybe a simplistic way to think that it's only due to immobilization, and it's probably much more complicated than just immobilization, and especially because even the non-radiative de-excitation, it's not really due to internal rotation; it's more due to photoisomerization of the chromophore. But so it could be also. Yeah, you're right. Yeah. Thank you.